Hello and welcome to our channel. In this video, we will be discussing about the tumor-inducing plasmid. So if you are new to our channel, subscribe the channel and press the bell icon. So let's talk about this TI plasmid, that is the tumor-inducing plasmid. So this TI plasmid, this was first discovered in the bacterium that is known as the Agrobacterium tumefaciens. So this is the tumor-inducing plasmid here, and the size of this plasmid is about 200 kb size. So this plasmid has the size of 200 kb, and this agrobacterium, this infects plants and it causes the formation of tumors that are known as the crown galls. So there takes place the formation of the crown galls. Now this TI plasmid, that is the tumor-inducing plasmid of agrobacterium, this is actually responsible for this tumorigenesis, formation of the tumors, the crown gall tumors. Now talking about the structure of tumor-inducing plasmids, so it includes the tDNA, that is the transfer DNA. So it includes the tDNA having the size of about 15 to 30 kb of size. So the size of the tDNA is 15 to 30 kb. So this portion is called as the tDNA. When we look at this tDNA, it contains the different types of genes like the auxin genes, the cytokinin genes, and the opine genes. So there is the presence of three different types of genes: the auxin gene, and then the cytokinin gene, then the opine genes. It is actually having the right border sequence and the left border sequence. This right border sequence, this is actually the sequence that is important in transferring the tDNA. Now after that, when we look into this tDNA region, this tDNA contains the left border inverted repeat, then here we have the right border inverted repeat. So this is the auxin gene, this is the cytokinin gene, and this is the opine synthesis gene. So here we can describe this tDNA region like this. So this is the tDNA region. This region is covered with the left border sequence and the right border sequence, and in between these two sequences, the invert repeats, the border sequences, we have here the three types of genes: auxin synthesis genes and the cytokinin synthesis genes, and then we have the opine synthesis genes. Now this auxin gene, when we zoom out this auxin gene, this contains the here the gene that is TMS1 gene, and then we have the TMS2 gene. So this TMS1 gene, this is for the tryptophan 2 monooxygenase formation, and this TMS2, this is for the indole estamide hydrolase. So these lead to the formation of the auxin, that is the phytohormone, and then we have here the cytokinin gene. This cytokinin gene, it contains the TMR gene, that is for isopentyl transferase. And when we look at the opine synthesis gene, so this opine synthesis gene here, it contains the OCS synthesis gene and the NOS gene, that is for the formation of the octopine and for the formation of the nopaline. Then we have the virulence genes that are called as the vir genes. So we call them as the virulence genes. So here we have the virulence region. So this is the vir gene operon basically. So here we have the vir gene operon. This is commonly known as the vir operon. It contains many types of the genes. in one operon. Then we have the opine catabolism genes. So basically we have seen the opine synthesis gene in the tDNA region, and now we are seeing the opine catabolism gene. So this is present outside the tDNA region. And here we have the origin of replication. So these opines, these are actually the nitrogen source, and these can be classified into about 9 different types of compounds, including octopine and the nopalline. So thus Ti plasmids, they are actually often referred to as octopine or nopalline type plasmids, depending upon the opine they specify. So what type of opine this Ti plasmid specifies, accordingly we name it as whether the octopine Ti plasmid or the nopalline Ti plasmid. So these are excreted from plant, and these are taken up by the Agrobacterium tumefaciens bacteria, and we have here the encoding region on the Ti plasmid that is involved in these opine utilization. So this was all about the Ti plasmid.